We are talking about genome alterations, mutation, and epigenetics. Epigenetics is at the end, and we're only going to have a couple of slides for it because we've already talked about epigenetics. We've had, I think, 2 or 3 chapters have talked about epigenetics now, so we should have a good idea of what that is and how that works. But mutations are heritable changes in DNA. They can occur in any body cell. In somatic cells, they're passed on by cell division to daughter cells. They're not transmitted to future generations because you only have one type of cell in your body that gets transmitted to future generations, and those are your germ cells, your sperm and your eggs. So if a mutation happens in your germ cell, your sperm or your eggs, then it gets passed on to future generations. It can be caused by environmental factors. We're going to talk about a lot of environmental factors that could potentially cause mutations that could be passed down to your children. Good morning, Jordan. Good morning, Priscilla. I think I said good morning to everybody. Methods used to determine whether abnormal phenotypes are caused by genetic disorders. Our pedigree analysis, we had a whole chapter about pedigrees, used to reveal potential inheritance patterns. You can tell if a disease is dominant, if a disease is recessive, if a disease is X-linked. You can also calculate mutation rates, do a little bit of math. The number of mutated alleles per locus per generation. Some diseases and disorders are only found at one location. We talked about sickle cell. We'll talk about sickle cell again. That is the mutation of one single nucleotide that changes an entire genome. Some, like cystic fibrosis, which we'll see, have a lot of different locations on chromosomes. So pedigree, just to review, an accurate pedigree information can be used to identify the individual who had the original mutation. This pattern of inheritance is autosomal dominant. There are no carriers here. There's no half and half circles. And it looks like there's about an equal number of men and women that get this mutation. Whatever this disease was. This is just an example. And you can see generation 1 didn't have it. So the mutation happens somewhere in generation 2. Generation 2, child number 5, which is a daughter. There are X-linked recessive traits that happen by mutation. New X-linked mutations can often be detected by examining the inheritance of the traits in males. The royal family is actually a really good pedigree to study because hemophilia runs in that family. And Queen Victoria, back whenever she was queen, seems to be the first one that had this mutation, that had this disorder, and passed it on to a few of her children. So one of her sons had hemophilia, and then 2 of her daughters were actually carriers. And you can see in generation 4, her and her husband, Prince Albert, had a lot of kids. They had 1, 2, 3, 4, 5, 6, 7, 8 children. So out of those 8 children, 3 of them possess the alleles to pass them on. This is just an expanded pedigree of the royal family. Like I said, there's Albert and Victoria. Victoria was the first known to have hemophilia. So she is where the mutation started, most likely. And then there's her children in generation 4, but you can see generations 5 through 8, all the males were all affected. And there's a few female carriers. And those female carriers pass it on to their sons. Mutation rate is the number of affected alleles in each generation. They're affected by gene size. Some genes are only a few base pairs long. Some genes are hundreds of base pairs long. Now, the presence of specific nucleotide repeats, or spontaneously or environmentally induced chemical changes. Skin cancer is an environmentally induced chemical change in your skin. That's how people get it. Part of it is genetic. You can inherit those genes from your parents. But if you spend too much time, if you have just the right genetics, you have just the right genes, and you spend too much time in the sun tanning or whatever, you don't cover up with sunscreen. And this is a lot for lighter skinned people. You don't see a lot of skin cancer in Mexican and Black people, or even Asians, really. There's just a table of mutation rates for selected genes. Achondroplasia is really high up there. That's dwarfism. Mutants per million gametes. There's 10 mutants per million gametes. They calculate that out to a mutation rate. Muscular dystrophy is another one that has a lot. Spontaneous mutations can result from errors in normal cellular processes, such as DNA replication. Can be caused by natural changes in the molecular structure and the bases of DNA. There's also induced mutations. Can be caused by mutagens, such as radiation and chemicals that affect DNA and cellular processes. And your Ed puzzle this week is about a specific mutagen that induced a lot of mutations in Vietnam over the war in the 60s and 70s. There's some errors in DNA replication. Mutations occur when DNA polymerase incorrectly reads the nucleotide on the template strand and inserts the wrong nucleotide into the new strand of DNA. So we went over our steps for replication. The DNA unzips, the enzyme, RNA polymerase, directs the free bases to pair with the parent strand. There can be errors in this process, in this step. Small chemical changes in bases can cause mutations. Normal base pairing between A and T contains 2 hydrogen bonds. That's picture A on the left. That's your normal base pairing between thymine and adenine. After something called a tautomeric shift, produced by a normal and reversible change in T, hydrogen bonding with non-complementary bases is possible, resulting in T pairing with a G. This is the wrong pair. T's do not pair with G's normally. But in the next round of replication, the original AT base pair will be replaced with the GC base pair in one copy of the DNA. Radiation is a process by which energy travels through space. There's energy traveling through space all the time. There are natural sources of radiation called background radiation, like breathing. That is energy. We breathe in carbon dioxide. We take in that energy. We breathe out oxygen. I'm sorry, we breathe in oxygen. We breathe out carbon dioxide. My bad. That is just natural radiation that takes place. It just happens in the background. But we are also exposed to radiation that results from human activity, including medical testing. How many of you have had an X-ray? You have to put on that apron to protect your internal organs. You're never, ever, ever supposed to have an x-ray if you're pregnant, because that will cause birth defects in your baby. Nuclear testing, nuclear power, and consumer goods. You guys are going to do a little bit of research on radiation, natural radiation, medical radiation in your discussion this week. Radiation can cause biological damage at several levels. As radiation strikes molecules in cells, it creates charged atoms called ions. Such ionized molecules are highly reactive and can cause mutations in DNA. At the top, it says radiation as a mutagen. A mutagen is just something that causes a mutation. Sources of radiation that individuals are exposed to in the United States. The average dose is 360 mrem. That's just the unit they use to measure radiation. Just over 80% is background radiation. Radon is considered background radiation because it's a natural element occurring in the universe. Cosmic, terrestrial, internal. Internal is us, like I said, like breathing. Cosmic is just space. Terrestrial is in the earth. There's energy that gets transferred all the time. It's considered radiation. Then there's all the other stuff, x-rays, nuclear medicine, consumer products. Chemicals can cause mutations besides radiation. Mutagenic chemicals that resemble nucleotides and are incorporated into DNA or RNA during synthesis are called base analogs. One is an analog used in research called 5-bromouracil, which has a structure similar to that of thymine and can be inserted in place of it. So if you get exposed somehow to 5-bromouracil, there is a chance it could replace a thymine in your genome. And when your genome gets replicated, it gets replicated with it and causes problems because it's not supposed to be there. Another base analog you guys are going to be looking at, like I said, in your Ed puzzle, it's called Agent Orange. It was used to spray and kill trees in Vietnam. So once inserted into the DNA, 5-bromouridine has base pairing properties of cytosine. In the next round of DNA synthesis, it will serve as a template for insertion of guanine in the newly synthesized strand. So what used to be an AT pair is now a 5-BRG base pair. And then after one more round of replication, the result goes from AT to a GC mutation. And it only takes that one mutation, like in sickle cell anemia, just that one base pair to cause problems. Chemical mutagens can modify bases in DNA and alter the base bearing properties. Some mutagens do this directly by changing one base into another. So it doesn't even have to be replaced. It can just induce base pairs to change. Treatment of DNA with nitrous acid changes cytosine into uracil. All you got to do is remove one amine group and cytosine gets switched into uracil. It's very simple. So what was a GC base pair is converted into a GU base pair. Uracil has the base pairing properties of thymine. So in the next round of DNA replication, the template strand containing a U will direct the insertion of an A into the new strand. And then after another round of replication, the GC becomes an AT mutation. So this is just the example. You have the GC on the left on the original, gets mutated into GU. And then after replication, the left strand, or the molecule on the left is normal, but the molecule on the right now has an A in the new strand because there's a U in the mutated strand. And then after another round of replication, that AT is there. That should still be a GC. Nucleotide substitutions, replacement of one or more nucleotides in a DNA molecule with other nucleotides. So the ones we've just been talking about are nucleotide substitutions. There can be a missense mutation where a single amino acid gets changed. A sense mutation changes a termination codon into an amino acid. So instead of stopping, the processes of transcription and translation just keep going. Or a nonsense mutation, where changes in amino acid get changed into a stop codon. That will stop translation too early, and whatever protein being made doesn't get made, or doesn't get made properly because it's too short. So missense mutations are like sickle cell. We went over this either last week or the week before. HbA is the hemoglobin molecule. At the top, you have normal HbA. It should be CTC, which pairs with a GAG and makes glutamic acid. But when a person has sickle cell, that T got mutated into an A and now makes valine. And then there's a couple different ones that could happen also. The original C gets put into a T. The original T gets mutated into a G. They all cause different problems. Frame shift mutations are insertions or deletions in one or more nucleotides. After the alter the reading frame, it changes the reading frame, which changes the amino acids in the protein. So this is a common example to illustrate frame shift mutations. You take a sentence made of all 3-letter words. The fat cat ate his hat. A nucleotide, in this case A, is inserted, the second codon, which destroys the remaining message. So if you insert that A into that sentence, you can see but the reading frame shifted. You can't tell what the rest of the sentence was supposed to be. And then a deletion will have the same effect. You delete the second codon, everything shifts to the left, and the sentence still doesn't make any sense. So these are called frame shift mutations because our codons are read by groups of 3. If you insert or delete something, that frame shifts and no longer makes sense. So this is just another example using actual DNA and amino acids instead of words. So an altered reading frame, when A gets inserted into the sequence, the codon's all shifted, and all of the subsequent amino acids are changed. So that middle amino acid is supposed to be tyrosine, but an A got inserted somehow and changed that first one to leucine, the second one to leucine again, the third one to glutamic acid. So the whole rest of the protein is messed up because the amino acids got shifted. Mutations can involve sequences called trinucleotide repeats. These sequences of 3 nucleotides are repeated several times in consecutive order within or adjacent to a gene. Mutations expand the number of repeats within a gene, converting a normal allele into a mutant allele, a phenomenon called allelic expansion. So if you add these repeats, there's more nucleotides, you expand the alleles. Fragile X syndrome is an example of allelic expansion. So it was first observed in the X chromosome gene FMR1, which is associated with Fragile X syndrome, the most common form of hereditary intellectual disability. The name of this condition derives from the observation that the X chromosome carrying the mutation has a stalk at the end of the long arm to which the chromosome tip is attached. So you can see in the picture, there's an X chromosome, and at the bottom, there's some extra stuff that shouldn't be there. That's a group of trinucleotide repeats that expanded the alleles and caused intellectual disabilities. This is just an example. Instead of under a microscope, there's the FMR1 gene. It's normally 6 to 52 copies of a CGG repeat, but a mutant allele contains more than 230 copies of that repeated CGG. There's disorders due to expanded trinucleotide repeats. Huntington's disease is one of them. Fragile X, myotonic dystrophy, has to do with your muscles. Mutations can be repaired. Our body normally does this for us. More than 175 genes in the human genome are dedicated to repairing DNA. So our body knows sometimes it makes mistakes, so it has a built-in system to correct those mistakes. Because our genome has a high rate of mutations, so many things can cause mutations in our genomes. However, sometimes that system gets overloaded and things like chromosomes becoming dormant, so they don't replicate anymore. It's called senescence. Apoptosis is cell suicide. Our cells will automatically commit suicide if something is wrong with them and just destroy themselves so it doesn't spread. Or if this doesn't happen, if it doesn't become dormant or it doesn't commit suicide, that's when cancer happens. The repair system is overloaded and escapes normal controls of the cell cycle. And it just keeps dividing and dividing and dividing, even though there's a mutation within that cell. That's when cancer happens. And the next chapter is actually all about cancer. During DNA replication, DNA polymerase adds about 20 nucleotides per second in the newly synthesized strand. Once in a while, the wrong nucleotide is added, like we talked about, produces the potential mutations. However, DNA polymerase can proofread its own work. Like you guys should all do when you're writing a paper. You should go back and read it and make sure you wrote everything correctly. If an incorrect nucleotide is added to the new strand, the enzyme can detect the error, reverse direction, and remove the incorrect nucleotide. It's that simple. Oops, I made a mistake. Let me go back and erase it. The enzyme then inserts the correct nucleotide and moves on. It's like when you're writing, especially in pencil, you spell a word wrong, you erase it, you spell it right, and you move on. DNA polymerase in our cells, they do this for us all the time. Because a few replication errors can escape the proofreading system, cells contain another system for detecting and repairing these mutations. Improper base pairing distorts the structure of DNA, and repair enzymes scan and recognize these distortions. Other enzymes, not polymerase. Once the distortion is recognized, these repair enzymes just cut the DNA on either side and remove the strand. DNA polymerase just fills the gap, and another enzyme closes the sugar phosphate backbone. So we have several built-in mechanisms to fix our DNA if something goes wrong. Like I said, they don't always work. But 99.9% of the time, our cells do just fine without us. Several genetic disorders, including Fanconi anemia, ataxia telangiectasia, and xeroderma pigmentosa, abbreviated XP, are caused by mutations in the DNA repair genes. These are when people's genes don't repair themselves after a mutation. XP is an autosomal recessive disorder with a frequency of 1 in 250,000 individuals. With 7 billion people on the planet, 1 in 250,000 is fairly common. Those affected with XP are extremely sensitive to sunlight. Sunlight is a UV light. Even short exposure to the sun causes dry, flaky skin, pigmented spots that can develop into skin cancer. Skin cancers are about 1,000 times more common in people who have XP. And it usually results in early death if these people don't take extraordinary measures, slather themselves in sunblock, stay out of the sun almost completely. This is probably where the tale of the vampire came from. People who really couldn't go out in the sun without getting seriously injured. Sickle cell anemia is due to a specific single nucleotide substitution, like we said. But then you have disorders like cystic fibrosis, which have more than 1,900 mutations of several types have been identified in the cystic fibrosis gene. It's abbreviated CFTR for cystic fibrosis. Trinucleotide repeat. This creates various symptoms in different patients. So not every single person with cystic fibrosis will have the exact same symptoms. Many different mutations have been identified, including deletions, nucleotide substitutions, and frameshift mutations. So that's the CFTR gene. And there's all kinds of different mutations in lots of different locations, which causes, like I said, a variety of symptoms, depending on where the mutation is specifically in that person. Epigenetics. Like I said, there's only going to be a few slides because we should all know what epigenetics is by now. These are heritable chemical modifications to DNA and its associated proteins that alter gene expression without changing the nucleotide sequence. The epigenome may change many times over a lifespan due to environmental conditions. DNA winds around the histone complex to form the nucleosome. Promoters that are wound tightly in a nucleosome are unavailable for RNA polymerase to start transcription. Chemical modification of the histones loosen the DNA, making that promoter available for transcription. Remember, the promoter is the start site. If DNA polymerase or RNA polymerase can't get to the start site, then it doesn't start. If it doesn't start, then those proteins, whatever they are, don't get translated. They don't get made. Depending on what the protein is, your body needs it. Your body doesn't produce things it doesn't really need, so. Oops. In both rats and humans, stress activates the hypothalamus region of the brain, which stimulates the pituitary gland to signal the release of stress hormones. So your body releases hormones to help you deal with stress. And that's the process. Those hormones are called glucocorticoids, and they come from the adrenal glands. So it's a whole pathway, like we talked about pathways last time. You are under stress, the hypothalamus area in your brain triggers the pituitary gland, triggers the adrenal gland to release these glucocorticoids. You have specific receptors for the glucocorticoids. We abbreviate those to GRs. On the cells in your hypothalamus that sense the level of stress hormones and will signal the adrenal gland to slow the release of stress hormones, bringing the body back into balance. So once there's enough stress hormones for you to deal with whatever stress there is, whether it's physical, emotional, once there's enough to help you deal with it, then your body kind of sends an off signal. Okay, we're good with the stress hormones, stop releasing them, your body goes back to normal. Well, they did an experiment with rats, because we always do experiment with rats. Rats with nurturing mothers that would feed them and care for them and nurture them, not only had more receptors, but also 90% less methylation in the GR gene promoter, which means that the gene is transcribed at higher rates. And if the gene gets transcribed, then that rat had a healthy stress response. But if these rats were not nurtured, then the opposite happened, and the rats didn't know how to deal with the stress. Their body got overloaded. So then they correlated this with humans. Child abuse and other early life hardships increase the risk of depression and suicide. So they wondered, can epigenetic changes be involved? So to see whether differences in maternal care in humans cause changes in the GR gene, just like it did in rats. Samples of the hippocampus were obtained after death in 3 groups. Suicide victims with a history of child abuse, suicide victims with no history of child abuse, and then victims who died suddenly but not by suicide. What they found was high levels of methylation were found in the GR promoter in suicide victims with a history of child abuse, but not in the 2 other groups. So we've learned in our epigenetics, if our DNA is highly methylated, it's squeezed really, really, really tight around the histones, it can't get transcribed. So those receptors don't get made. The results were consistent with those in the rat, suggest that epigenetic mechanisms might respond to the social environment and alter the stress response later in life. So people who live with abuse, they don't have a good stress response because their hormones, they aren't received properly. Because in their DNA, those histones are methylated, can't get transcribed, can't get translated, and those proteins that make the receptors to receive those hormones don't get made. So their stress response is very, very different than somebody who had nurturing parents. I think, yep, that is our last slide. Questions? No?
No, I don't have any questions.
Okay, thank you. Then we are good for the day. Like I said, you guys have an ed puzzle on Agent Orange and the horrors that our government committed, again, because that's all they seem to do in the past, or they still do sometimes. You have another assignment. You have a discussion. You're going to be researching radiation and some mutagens. And then your practice questions and your test. So that's it. You guys can log off.
All right, bye.
Bye, Shay.
Bye.